Hello friends, this video on respiration in plants part 5 is brought to you by examfear.com. No more fear from exam. About this. So now it is time to talk about aerobic respiration in detail. So let us try to understand the steps of aerobic respiration. As I said, it is a multi-step process. The beginning is glucose and the end is carbon dioxide, water and a lot of energy in the form of ATP molecules. So let us see what are those steps which will actually make this conversion happen. So the entire process of respiration, right now we will be talking about the aerobic respiration. It has 4 steps: glycolysis, pyruvate oxidation, Krebs cycle and electron transport chain or electron transport system or ETS, whatever you call it. So these are the 4 steps which together will lead to the conversion of glucose into carbon dioxide, water and a lot of energy. So first we will start our discussion with glycolysis. So now this step, that is the first step of respiration, glycolysis, is a common step to both aerobic and anaerobic respiration. So in aerobic as well as anaerobic, the only difference is the presence or absence of oxygen. So the first step, that is glycolysis, it does not require oxygen. That's why this is the first step in case of anaerobic respiration also, right? So what comes to your mind with the word glycolysis? Glyco, it is something related to glucose, glycogen, something like that. So glyco would mean sugar. Lysis, what is the meaning of lysis? It means to split or splitting. So glycolysis means splitting sugar, and that is what we are going to do. Sugar here, as I said, the starting material of our respiration process is glucose. So we are going to split glucose. We are going to break down glucose into simpler components. So that is what we will do in glycolysis. So it is the process in which glucose is broken down into 2 molecules of pyruvic acid. So glucose will be broken down into 2 molecules of pyruvic acid. So what is glucose? Glucose is a 6 carbon compound. There are 6 carbon atoms present in glucose. But when you look at pyruvic acid, it is a 3 carbon compound. So there are 3 carbon atoms in 1 molecule of pyruvic acid. So glucose will be broken down into 2 pyruvic acid molecules. That is the agenda of glycolysis. So this is the glucose and this is your pyruvic acid. So if you see, this is a 6 carbon compound, 1, 2, 3, 4, 5, 6, and it forms 2 pyruvic acid, 1, 2, 3, again 1, 2, 3. So glucose will be broken down into these 3 carbon compounds. Now this glycolysis is also known as EMP pathway. Why is this called EMP pathway? From where did it get EMP? E stands for Emden, M stands for Meyerhof and P stands for Parnas. So Emden, Meyerhof and Parnas, these were the 3 scientists who together described this scheme of sugar splitting for the first time. So, or in other words, you can say that these 3 scientists together described glycolysis as a process for the first time. And that is why after the initials of their names, it is known as EMP pathway. Now this is again a multi-step process. So glycolysis itself is just the first step of the process of respiration, and this glycolysis itself is also made up of 10 steps. So just imagine how many steps are involved in the entire process of respiration. So now our job is to understand the 10 steps of this process of glycolysis. Now before we start understanding the steps, the question is, where does glycolysis take place? So the question is, where does glycolysis take place? Now glycolysis, the process will start with glucose. So let us first try to see where is glucose formed in a plant. So now this is your plant. So where does photosynthesis take place? It takes place in the leaves, right? So if photosynthesis takes place in the leaves, that means glucose is formed in the leaves, but glucose gets transported to different parts of the plant with the help of the phloem tubes. Right? So the phloem tubes will transport the glucose to different parts of the plant. When I say it is transported to different parts of the plant, I mean to each and every cell present inside the plant body. So basically the glucose will reach each and every plant cell. So the glucose will enter the cytoplasm of the plant cell. So this part of the plant cell, glucose needs to be broken down into simpler form. So that's why glycolysis will take place in the cytoplasm of the plant cells. And this is the first breakdown of glucose. So we say that glycolysis occurs in the cytoplasm of the cells. So this process of glycolysis will take place inside each and every cell of the plant. So now let us look at the 10-step glycolysis process. We will start with step 1. So in step 1, in each and every step, now since we can understand that since it is a 10-step process, that means a lot of intermediate products will be formed, because at each step some product will be formed, right? So a lot of intermediates will be formed. A lot of enzymes play crucial role in each and every step. So we will talk about the starting material of each step, the product of each step, and the enzyme which makes that step take place. So we'll talk about these three things. So for step 1, the starting material is obviously glucose. Okay. Which enzyme participates in step 1? There is an enzyme called hexokinase. And what is the product? The product is glucose 6-phosphate. So basically in this step, the 6-carbon glucose. So glucose is a 6-carbon compound. So this 6-carbon compound will get converted into glucose 6-phosphate. So what is glucose 6-phosphate? That means on the 6th carbon, a phosphate group is present. So 1, 2, 3, 4, 5, 6. This is the 6th carbon, and on the 6th carbon, a phosphate group is present. Right? And how it happens? It happens in presence of the enzyme called hexokinase. And during this step, ATP is utilized. Now, the question is, from where do we get this extra phosphate? So that phosphate comes from ATP. So ATP will be utilized in this step. What is ATP? ATP is adenosine triphosphate. Triphosphate, that means there are 3 phosphate groups present in ATP. So this ATP in presence of this enzyme hexokinase will get converted into ADP, that is diphosphate. So 2 phosphate groups will be there. So what happens to the other phosphate group? So that extra phosphate group from ATP comes to this compound glucose, and that is how it forms glucose 6-phosphate. So that is how glucose 6-phosphate is formed. So in this step, ATP is utilized. So when we say ATP is utilized, that means energy is utilized in this step. And what is this process called? The process where inorganic phosphate is added. That process is known as phosphorylation. Phosphorylation. So adding an inorganic phosphate group is known as phosphorylation. So let us look at the next step, that is step 2. So what would be the starting material for step 2? Obviously the product of step 1. So the starting material here would be glucose 6-phosphate, because this is what was formed in step 1. So in this step there is an enzyme called phosphoglucoisomerase. Now this, whenever you have an enzyme whose name ends with isomerase, that means that enzyme helps in changing or it helps in rearranging the molecular structure of a compound to convert it into its isomer. That is why the name isomerase. What are isomers? Isomers are those compounds which have the same molecular formula but they have different structures. So that means if there are 6 carbon atom in glucose 6-phosphate, its isomer will also have 6 carbon atoms, but the way they are arranged will be different. So isomerase enzymes always help in converting one compound into its isomer. So what would be the product here? The product would be fructose 6-phosphate. So now glucose and fructose, if you compare the structure of glucose and fructose, this is glucose and this is fructose. So if you look at both of them, both have 1, 2, 3, 4, 5, 6 carbon atoms. So here also it has 1, 2, 3, 4, 5, 6. Again, if you see, the number of hydrogen atoms as well as oxygen atoms are also same in both of them. So their molecular formula are same, but the way they are arranged is different. So that is why glucose and fructose are isomers. Now, due to different arrangements, many of their properties also vary. And that is why they are two separate compounds. Otherwise, they would have been just the same thing. Right? So in this case, glucose 6-phosphate will get converted into fructose 6-phosphate. So here what will happen? So the starting material is also a 6 carbon compound, something like this, with a phosphate group at the 6th carbon atom, and the product will also be the same, a 6 carbon atom. It is just that the arrangements will be different. Now I am not getting into the arrangements here. I am just trying to tell you how many carbon atoms and how many phosphate groups will be there on both sides of the equation. The next step, your starting material will be the fructose 6-phosphate, that is the product of step 2. So here fructose 6-phosphate and the enzyme is phosphofructokinase. So what will this enzyme do? This will convert it into fructose 1,6-bisphosphate. Let us see how it happens. Now whenever you have enzymes with kinase, they generally make phosphorylation take place. See, the first enzyme was also hexokinase, there also phosphorylation took place. Here also we will see what happens exactly. So here we start with fructose 6-phosphate. So 1, 2, 3, 4, 5, 6. So this is fructose 6-phosphate. It has 6 carbon atom and 1 phosphate group, right? So in presence of this enzyme phosphofructokinase, what will happen? It will again utilize one ATP. So this ATP will get converted into ADP. So triphosphate will become diphosphate. So that extra phosphate group will come and add on to this reactant. So as a result, the carbon atom you have is still 6, but now you will have 2 phosphate group. One was already existing and the other one will come from this ATP. So it becomes, so one phosphate group was already here and the other phosphate group gets added to the first carbon atom. Therefore, it becomes fructose 1,6-biphosphate. So if this is 1 and this is 6, so 1,6-bisphosphate. So this is the product here. So here also if you see phosphorylation took place because inorganic phosphate was added to the compound. So here also phosphorylation takes place. So here again 2 molecules of ATP are utilized in this step. So here also ATP will get utilized. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.